Hey all, so today we're going to be talking about D3.2 inheritance, and here we're going to be looking specifically at pedigrees. So we need to be able to answer the questions, what patterns of inheritance exist in plants and animals, and then what is the molecular basis of inheritance patterns? And so we're looking at page 699 to 706 in the Oxford textbook, and we're thinking about continuity and change. So single nucleotide polymorphisms are related to small changes that occur in genes, or so it could be in coding sections of genes or non-coding sections of genes. And these are going to cause alleles. And then the gene pool is all the individuals reproducing or able to reproduce in a population. So if we have different bases in a gene segment, then those are going to be called SNPs, single nucleotide polymorphisms. There can be many, and then that means that there can be many different alleles. And each individual receives two alleles from their parents, one from mom, one from dad. And for example, the S genes for apples, there are 32 different variants, and that prevents self-pollination in apples if any of the pollen is the same allele that it already has, because it's just too close then for it to be reproducing with itself. And ABO alleles are examples of multiple alleles. So those are 4 different alleles. And A and B are codominant, meaning they will both show up if you have those alleles. And O is recessive, and that's going to result in 6 possible genotypes, but only 4 phenotypes. So the 4 phenotypes are obviously A blood, B blood, O blood, or AB blood. But there could be 6 different genotypes. So we'll talk about this in just one second. So firstly, I stands for immunoglobulin. That means antibodies. And because this is a codominant trait, we need to write A as a capital letter. So it's going to be a superscript with this I letter, and same thing with B. And then O is going to be recessive, so it's a small letter I. And so if we both of our alleles are A alleles, then we would have A blood. And if we had A and B alleles, then we would have AB blood. So we can see that those two there are the same. And if we had an A allele and an O allele, we would still end up with A blood, but we would have a different genotype. And so it would be this capital IA and then small I. So we'd have an O allele and an A allele. And we can see that that one and that one down there are the same. And same thing with these Bs. So if you had B blood and an O allele, you would have B blood, but you would have a genotype of essentially BO gene alleles. If you had 2 B alleles, you'd end up with B blood. And if you had 2 O alleles, you would end up with O blood. And so those are your 6 different genotypes. And so if you have B blood, if you have A blood, it's going to make anti-B antibodies, which means that you will have antibodies in your blood that combat B blood. So if you got a blood transfusion, then your blood would clot and you would die because those antibodies would attach onto the B antigens. And if you had B blood, you would make anti-A antibodies. And so if you have O blood, you are the universal donor, which means that you are your blood cells have no antigens, so they can go in anywhere, but you make all of the antibodies. So you can only get O blood because your O blood has no antigens. And if you have AB blood, you have all of the antigens, and so your body makes none of the antibodies. And so you are the universal receiver of blood. All right, so incomplete dominance is kind of similar to codominance in the way that we think about it, but incomplete dominance is where white flowers, which make no pigment, and red flowers produce a lot of red pigment if both of the alleles are red. If only one allele is red, then it's making some red pigment, and that results in pink flowers. And so also one thing that we need to think about here is polygenic traits. So that means many genes are controlling for this trait. And so here we have a graph of skin color. And so there are a lot, 6 different genes that are controlling for skin color. And so we have this normal distribution, a bell curve happening because of it being a polygenic trait. And blood type is not a polygenic trait. So it's just one segment on a gene. And these are going to be discrete categories. So like you either have A blood or you have B blood, you have AB blood or you have O. Those, there's no like kind of merger of that. But a lot of human traits and animal traits are polygenic traits. So like things like height or intelligence level, how do we even quantify that? Like skin color, like foot size. So all of those things, like there's a range of people that have bigger or smaller feet or darker or lighter skin. And so that is a key indicator that you are talking about a polygenic trait, if you can see that normal distribution happening. And recognizing with the skin color gene, the darker that your skin is, the more protected, the more melanin you make, and the more protected you are against UV light. So this was a highly adapted feature in humans who had high sunlight conditions, and it was evolutionarily advantageous for them not to get skin cancer. All right, so sex-linked traits. This was discovered by Thomas Morgan in the 1910s with fruit flies. So eye color is red or white in fruit flies, and it's on the X chromosome. And so the X chromosome has about 9, and this is in humans. The X chromosome has 900 genes, and which are important to males and females. And the Y gene, this is again in humans, this is not fruit flies. So with Thomas Hunt Morgan, his fruit fly thing, what he was doing was he was seeing that some male flies were producing certain eye colors, and that was only occurring in the male flies. And so that was how he discovered sex-linked genes. So going back to human chromosomes, X and Y chromosomes, the Y chromosome only has 55 genes, and these are specific to males. And so the SRY gene is on the Y chromosome, and that's going to trigger testicle growth, which will make testosterone to grow other male organs when the fetus is developing. If there's no SRY gene, then the default fetus is female. So if a person is male and they have a faulty X chromosome, so allele, then they may have no backup allele on the Y chromosome, because the X and the Y chromosome are not homologous, meaning they don't have the same genes at the same loci throughout. So when women have the disease, they would have to have 2 faulty alleles. So for a woman to have hemophilia, she would have to have received a hemophilia allele from her dad and a hemophilia gene allele from her mom. And that would be the only way that a female would have it. But in the case of a male, which has an X chromosome and then a short Y chromosome, then the faulty chromosome comes from the mom, and the dad produces the Y chromosome. And so then that human would have only one allele, and they would have hemophilia. So in this case, this is for hemophilia here. And so we can see if the mom is a carrier for hemophilia and the dad does not have hemophilia, then in this case, we would have a 25% chance of the children having hemophilia, but a 50% chance of the male children having hemophilia. All right, so this is a pedigree chart. You've probably heard of pedigrees because of dog breeding, maybe. But there's over 4,000 genetic conditions, but the risk of having 2 alleles for the condition is rare. On average, each person is a carrier for about 5 rare recessive alleles. So it's really good if you don't reproduce with close family members so that you're not passing on the same 5 rare recessive alleles that you both have because you're related to each other, which is why you don't have children with your first cousins. So pedigree charts can show inheritance patterns and show the genotypes of the parents. And so this is going to depend on what genetic condition you're investigating. So how it presents is going to be different. So if it's sex-linked, it's usually going to show up mostly in males. And so you can see here, this parent combo had 2 normal male children. And so potentially it's not sex-linked. The female child had it. But then when we look up here closer, this is a male with hemophilia and a normal female. But so we have the carrier is being produced here. And so this was quite lucky, and the Y chromosome here is going to be passed on from the male and produce male children. So this doesn't always mean that sex-linked will show up in the pedigree chart always in males. So we have to be careful when we're looking at it. And so in this case, we have the male has hemophilia and the female is a carrier. And so we have 1 hemophiliac male, 1 normal male. Remember, it's a 50-50 chance if you have a carrier female, and then a carrier female, and then actually a hemophiliac female, because that's coming from the dad's side and the mom's side. If you're looking at a genetic condition that is recessive, the parents will not have the disease. Pedigree charts don't always show if someone is a carrier. They usually show if someone has the disease or does not have the disease. And so in the case of a pedigree chart where the condition is recessive, you would have both parents are carriers, but they don't have the disease. And then you can see the disease showing up in the children. And that's an equal split between male and female children. And then if it's dominant, 1 parent has it, but then multiple children can have it. So up to 50% or approximately 50% of the children could have it, both males and females. And so this here is a sex-linked trait. We know that hemophilia is a sex-linked trait, but in this case, we have a normal male and a carrier female. And so what we can see here is that normal male, that's actually a marriage in, sorry, a partner in. So of the males, 50% of the males have the disease, and none of the females have the disease in this original pairing. And so that could be an indicator that this was a sex-linked trait. And then again, we see it here where we have 50% of the males being hemophiliac, as well as one of the females. But in this case, we have a hemophilia dad and a carrier mom. So to summarize, codominance is when both alleles will show up in the phenotype. So for example, AB blood. Incomplete dominance is when some of the trait is showing up, but not fully. And then polygenic traits are controlled by multiple genes, and this results in a continuous trait. So this would be something like height or skin color or potentially intelligence. And so sex-linked shows more in males because they don't have an extra X chromosome if one allele is different in a problematic way. And hemophilia is as well as red-green colorblindness are sex-linked traits. And then pedigree charts show inheritance patterns in families. And so here is a pedigree chart for albinism. So being an albino person or having very white skin without pigmentation. So looking at this situation, would you guess that albinism is a recessive, a dominant, or a sex-linked trait? I will leave that to you to figure out, as well as they have some question marks in here as to whether or not the what was the genotype of these people. So come talk to me if you can't figure this out in class, and we can go over it together. All right, thanks for watching. That's it for this video.